It's now my pleasure to introduce our first speaker for this virtual Discrete Math Day. So Megan Owen, I've gotten to know as one of the leading experts on understanding the space of phylogenetic trees, a concept which was sort of laid out in a really groundbreaking paper by Valera, Voltman, and Holmes. Megan did her PhD at Cornell underneath the direction of Luis Valera, graduating in 2008. She then took a very prestigious postdoc at SAMC, and then joined the faculty at Lehman College as part of the CUNY system some years later. And Megan has continued to write really fundamental papers in the analysis of Fréchet means and concepts related to variance on tree space, and was recently recognized by an NSF career grant, which she was awarded last April 2019. And so I am very excited to have Megan here, and even though it's virtual, and to tell us something about the analysis of tree space. So Megan, are you ready to take over and share your screen? Oh, sorry, you're muted, if you want to unmute yourself.
Yes, okay, so let me just do that.
Great.
So, okay, and just to double check, can people see my mouse pointer?
Yes. Yeah,
yes. Okay, great. Okay, so thank you very much to the organizers for inviting me, and thank you everyone for showing up to this virtual version of this. And also, I just want to please feel free to ask questions at any time during it. That's not a problem at all. So the work I'm going to be talking about is joint work with Jillian Grindstaff, who's a grad student at UT Austin. So I just want to give an introduction first about phylogenetic trees. So they show the evolutionary relationship between a set of species. So in this case, we have 4 fish, and we have some DNA sequences from the fish, and then we want to show which that these 2 fish are most closely related, right? And then next this fish, and then next this one. Okay. And the trees I care about have branch lengths on them. Now, usually the branch lengths are actually decimals less than 1, but it's kind of annoying to look at those in a talk. But they would represent sort of some kind of evolutionary distance between the nodes, so something like the rate of mutation. And they can represent time, but those, if it is going to represent time in a tree, it has to sort of be carefully calibrated with like fossil evidence, for example. And they're used in evolutionary biology, population genetics, epidemiology. So if anyone has been to nextstrain.org, they have a really interesting, they've done a phylogenetic tree of COVID-19 and sort of showing where the different outbreaks have sort of come from in the world. And they were also used for when they were trying to figure out where Ebola was coming from in Africa with the recent outbreaks. And then they're also used in oncology. This is a really recent, I guess, kind of technology sort of allowed people to sequence cells, because the cells mutate so rapidly that you can sequence the cells in a cancer tumor, you know, over the span of a few months or years, and you can actually get a tree showing how it's mutating. So I'm going to sort of, okay, so usually what we want is what's called the species tree, which shows how your different, say, species of fish are related. But the DNA actually gives gene trees. So for each gene, you want to look at how, so say for this blue gene, whoops. Okay, so say for this blue gene here, right, that's in all the fish, the evolutionary history of that particular gene has these 2 fish being most closely related, so similar to the species tree. But for this red gene, you can actually see that what happened here is in this ancestral population of fish, there were 2 variants of the gene, the one that went into this fish and the one that went into this one. And I mean, that's the same, that's just would be like two alleles, right? So just like right now humans have different versions of the gene for eye color, for example, right? So there's just two different versions of the gene in this population. And this version actually first has a common ancestor with this fish, actually, and then with this one, and then finally with this one. So the gene trees can differ from the species tree, which obviously makes it more complicated to figure out the overall species tree. So what I was just talking about is due to something, it's called incomplete lineage sorting. This idea that your lineages are not sort of, it's called coalescing, or like you're getting your ancestor in that sort of branch right above where the two populations come together. But you can also have things like horizontal gene transfer. So this is in, it happens a lot in bacteria, but it happens in some other cases too, right? So for example, some bacteria eat some other bacteria and absorbs some of the DNA. And so you're getting a DNA transfer across sort of between two lineages instead of just from the parents above it. And then finally, and this is the one that this talk is sort of addressing, or partly addressing, is you can have either gene duplication. So a gene at some point, somehow there ended up being two copies of it. And so, for example, this species only has the one copy here, and then species B would have both the copy in red and the original in green. And it's possible they could even have different functions at this point, or slightly different functions. And you can also have gene loss. So C actually just has only the copy of the gene. It doesn't have the original gene because it just got lost at some point. And so this is pretty common. And I guess I should say this talk is really just addressing the issue of loss. So if, well, and duplication. So essentially the fact is that your gene trees, especially now when you're sequencing, say, thousands of them for a set of species you're interested in, might not have all the same, exact same set of leaves, but you still want to be able to analyze them. So I'm going to think, so mathematically, how will I think about these trees is as what I'll call metric trees. So there's two parameters. There's the tree shape, which is called the tree topology. So if there are any actual topologists, I mean, I know this is not the correct use of the word, but this is just what this is sort of what biologists use this term a lot. But that's just talking about like the branching pattern, right? That these two trees are most closely related, and then this one, etc. So the combinatorics of the tree you could think of. And then the other parameters are the edge lengths, and they are Euclidean, right? Because they're just numbers. And you can actually, I won't talk about it here, but you can put instead vectors on the edge lengths. So if your tree was representing something like a like anatomical trees. So I've also done some work with like your lungs form a tree shape, and you can think of the branches as being like a 3D. You can put a vector to represent the 3D shape of it. So this does extend, but we're just going to talk about numbers, which would be the rate of mutation roughly. And so this isn't Euclidean data because of the tree shape. That's really causes all the problems. And as I will show you, if I look at all trees on the same, all possible metric trees on the same set of species, because the branches can change, the branch lengths can change continuously, I actually can get a whole continuous space. And that's what I will now explain. So this is the BHV Treespace Framework. It was constructed by Bolero, Holmes, and Vogtman in their 2001 paper, which Justin mentioned in his introduction. So as I said, it's a set of trees with end leaves and branchlets, and it does include what are called degenerate or non-binary trees. So the one over here is an example. So in a binary tree, every branch ends in two children, except for the leaves. And in a non-binary one, you can have, for example, this branch here has 3 children coming off of it. What else? I think that's it. Okay. So let me explain a little further. So biologists have this idea of splits. And what it is it's just an edge, and it's the partition that it induces of the leaves, right? So this edge E3 it's splitting the leaves into two sets, A and B, and then everything above it, 0, C, D, and E. I should say in this talk, all my trees are going to be rooted. So often I'll put a leaf 0 up here. This all works with unrooted trees as well. You just pretend the root is a leaf and it all works. But for this talk, I'm just going to use rooted trees. And the notation I'm going to use for the splits is just the set of leaves, one of the sets of leaves, a bar, and then the other set of leaves. So we can represent each tree as a vector, where the coordinates are the possible splits. So for example, first I have all the splits corresponding to the pendant edges, so the edges ending in leaves, right? And then the number here is how long that length is, right? So the edge with A split off from the rest of the tree has length 2, and so I have a 2 here. Okay, and then I have all other possible splits. So for example, this red edge, right, has AB split off from the root and CD. And that has length 2, so I would have a 2 in its position here. And then I don't have AC like split off from the rest, so that's a 0. And then I have my blue edge over here, which is CD split off from the rest. And then, so there are a lot of zeros. So I actually never, like there's code for a lot of the stuff I'll talk about. So you would never actually represent the trees this way in code because it would be very inefficient because there's an exponential number of possible splits. But as a way to like conceptually think about it, it's a good way. And so you can see in this tree, it's missing the blue edge, right? Because C and D just got a 0 in that position, but it has a 2 in the position for A, B, and C split off from D and 0. Okay. So this is looking maybe kind of Euclidean, right? I've just got to represent my trees as Euclidean vectors.
Okay.
But I told you.
Hello,
Yeah.
I have a quick question. Is
Yeah.
there one coordinate for each partition of the leaves?
Yes.
Okay, cool. Thanks.
Yeah. Yeah, so that's why there's like a huge number of coordinates, but most of them would be 0. Yeah, so why is this not actually a Euclidean space? And that's because we have this idea of split compatibility. So we say 2 splits are compatible if there can be a tree that contains both splits, right? So this tree, for example, contains both the split with AB split off from the rest of the leaves and with ABC split off from the rest. But I can't have the tree, F isn't compatible. I couldn't have A and C paired together and A and B paired together in the same tree. So I can't have both, like both those 2 as cherries, like that just wouldn't work because A would be in 2, like paired with 2 different leaves. So those 2 splits would be incompatible. So what that means, right, so in particular, if these 2 splits, AB split off from everything and AC split off from everything, are incompatible, it means they can never both be positive in the same vector. So not all vectors are possible. And our tree space is only those vectors that actually correspond to trees. And that's why it's not a Euclidean space, because we're missing huge chunks of like if we're thinking of this as I think it's roughly 2 to the n vector space. There's some little adjustment there, right? We're missing huge chunks of it. Okay, so I'm going to show you one other way to think about this space too, which is like a constructive approach. So I'm going to kind of build it up. So here we have our tree with its 2 edges, and I'm now going to ignore the edges ending in leaves, just because they kind of add a lot of dimensions, and they're in every single tree. So mathematically, they're not very interesting. You can kind of think of the part of the tree space that corresponds to the interior edges. The full tree space is that the tree space corresponding to the interior edges crossed a product with the just R to the N for your edge lengths, or edges ending in leaves. So I'm just going to ignore them. Yeah, so we have our AB here, CD, that edge here. And then this tree, right, it had the different, instead of C and D split off, it had A, B, and C. So it would be trees with different edge lengths would be different coordinates within this quadrant. So you can see how that works, right? So in this tree, right, the red edge is short. So I'm like over on this axis, right? And the blue edge is longer. So I'm up farther at this. So depending on those the lengths of these interior edges, that's going to tell you what coordinate they are within the orthant that corresponds to them. And then trees that are degenerate, so this one, right, only has AB, the AB edge, it's going to be on the axis. So you can think of like shrinking, shrinking like say the CD edge down to 0 to get this tree. If you just imagine that edge shrinking, you get from this tree to this tree. And then I can imagine growing out the purple edge and moving into this orthant. Okay, so let me just add in the rest of this isn't actually the rest of this is 5 or 3 more trees. So we can see that we now have 5 quadrants around an origin. And so that is definitely not Euclidean, right? Because in Euclidean space, we have 4 quadrants around an origin. And this is only 5 of the 15 total quadrants. And I'll show you how they all fit together in a second. But just to show you how do we compute distance in this space is that if I have 2 trees, so if I have any path between them, then I can compute, so this happens to be the shortest path between them, but I can compute, right, like each of these are orthants or quadrants. An orthant is just the n-dimensional version of a quadrant or an octant. Okay, so within the quadrant, right, it's Euclidean, so I can compute the length of the path, and I can add up the length of the path in each of the quadrants. So if my path went up like this, I could still compute its length. And then the geodesic is just the shortest of all possible paths. And here, because I can draw this picture, so really this picture should be sitting in 5 dimensions, 1 for each axis. But because we can't have negative branch lengths, I can kind of double up the axes. And you can see that I can think of kind of laying these ones down flat, and then it just becomes a Euclidean space, and it's obvious that this is the shortest path between the 2 trees. But if instead my 2 trees were over here, so these 2 have the same combinatorics, the same tree topology, right? The same tree shape, but different branch lengths. Same with these 2. But now the shortest path between them actually goes through the origin. And to see that, if you thought of instead, so you might think, well, like clearly it's not faster to go kind of bulge out like this, right? And so you might be, so if you're thinking, well, why can't I sort of cut this way? It's like if I actually lay out these 2 quadrants, you can see that it also doesn't work. So the combinatorics of the geodesic, which orthant it goes through, depend both on the tree shapes and on the branch lengths. So this is a space that kind of holistically combines both of them, which a lot of previous methods in phylogenetics, because I mean, obviously, biologists have thought about how to compare distances between trees and stuff before, but a lot of methods only dealt with sort of the tree shape, or if they brought in the branch edges, it was like not as intertwined as here. So, yeah. Yeah.
It looks like you're getting something hyperbolic. Is there any, I don't know, use in using like a hyperbolic metric there instead of the straight edges that you have?
Yeah, so this is hyperbolic. This is a non-positively curved space. I'm trying to think. I sort of feel like I've maybe seen a paper where someone did something like that, maybe with a different space, but I don't remember. I mean, possibly. I guess I'm not super familiar with hyperbolic spaces, so I'm not sure exactly how it would work, but possibly. But yeah, you're completely right that it is like non-positively curved.
Thank you.
Okay, so yeah, so I said these are only 5 of 15 quadrants, and so if you want to know how they all fit together, I can think of taking the link of the origin. So if you know what the link is, great. If you don't, just think of taking across each quadrant, I just take the line from 0, 1 to 1, 0, so the simplex, and they form the pentagon here. And if I looked at those lines for all 15 of them, I would get the Peterson graph. So that outside, for example, 5 is the 5 we had. And then I have my other 15 quadrants in here. And you can see that actually at each axis, there are 3 quadrants attached to it. And that's another sign this isn't Euclidean. Because again, that's impossible in Euclidean space. Okay, and then finally, one more way to think about geodesics is you can think of them as morphing one tree into another. So this is one tree going to this tree. And what I'm doing is I'm sort of shrinking down the branches in this one, and then I start to grow out new branches, and I kind of morph the one tree into the other by growing and shrinking branches. So somehow as you go along the geodesic, it's a minimum way to morph trees. Okay. So yeah, so as I sort of mentioned, Treespace has global non-positive curvature. And yeah, so hyperbolic space is negatively curved. Euclidean space has 0 curvature. The Earth or a sphere has positive curvature. So Treespace is non-positively curved because it has those sections. It has those Euclidean sections that are have 0 curvature, and then it has things like the origin that have like negative infinite curvature. And so this is called the cat-0 space, if you've heard of the term. What it means is that geodesics are unique. The geodesics are just the shortest paths between points. And so, for example, we have a well-defined midpoint tree. This is also telling us that the sum of this, like at least squares, let me think. If you find the minimum of the sum of point minimizing the sum of squares, there's only a unique one of those, so a unique mean. So it's got some nice properties, which is good. And these are also properties that Euclidean space has. So the BHV distance or geodesic distance is just the length of that geodesic or shortest path. And with Scott Proven, we had shown that you can compute this distance in n, so the number of leaves, times m cubed. And that's just saying that, so in biology, often you'll have splits in common between the 2 trees you care about. You can decompose the trees along these common splits, and then m is just the size of the biggest subtree you're left with, where there aren't common splits anymore. Basically, it means that if you have completely random trees, this sort of becomes n to the 4th, and it's maybe not as good of an algorithm. But if you're working on trees, like biological trees, where you expect them to have some relation to each other, then it actually is a pretty reasonable algorithm. And so there's also a lot of statistics on this space, which I mainly just mentioned, so you know it's out there. Things like how to compute mean, median, variance, central limit theorems, principal component analysis, confidence regions, other random sort of statistical techniques, and there's probably stuff I'm missing here. The only problem is that all of these methods assume that your set of trees has exactly the same leaves, which, as we just saw, is not guaranteed, right? We said that if you have a bunch of gene trees, say thousands of gene trees, you would expect that some of them are missing some leaves. And so we would like to have a way to kind of statistically think about trees with gene trees with missing leaves, and somehow be able to like analyze them or study them and kind of take care of that. Or perhaps to like figure out a way to, if we have these trees with missing leaves, to somehow still find the overall species tree, or a tree that can kind of accommodate them all. So obviously this is a huge problem. People have thought about it before. If you know the area of phylogenetics, it's often called the supertree problem. Or a supertree is this idea of taking trees with partial leaf sets and somehow putting them together to make your overall species tree. So this is kind of just one small contribution towards this very big problem that is still actually unsolved. And it's growing more and more important as we get better technologies for sequencing DNA, right? Because we can just get So back in the 70s, right, we didn't have these problems because it was so hard to sequence something. They would only pick one particular gene and sequence only that gene and say the species tree must match that gene. And now instead we're sequencing thousands of genes, and we want to somehow build the species tree, take all that information we have and put it into the species tree. So this will only sort of become a more important problem as time goes on. So I'm just going to think about this the problem we're going to think about here is if we have a tree with n leaves, which is a subset of capital N leaves, how can I extend the tree to that? And how can I think about these sort of extensions of trees? So there's a paper, Wren et al., which was by Ruth Davidson and a bunch of students she worked with, undergrad students she worked with, who gave a combinatorial framework for thinking about what sort of tree topologies are possible when you add the missing leaves. And we want to look at what happens with metric trees. Okay. So what I'm going to talk about is an extension space, or explain what that is. And that is this idea of like, I have my tree here, it's missing leaf 4. What are the possible metric trees I can get when I add this in? And those will form the extension space. So what I'm going to do is I can think of adding leaf 4 to this orange edge here. And so maybe let's go one more along here. So you can see it's making a new split 2, 4, right? And then I still have my green split here, 1, 3. And that's what these axes are, the 2, 4 split and the 1, 3 split. And so when this leaf was added right at the very tip, the 2, 4 split was actually 0. So we were here. Now the 2, 4 split was actually this whole orange line, so 0.3, so it was here. Now I've moved it a little along it, so it's now like, I don't know, maybe here, the tree I'm showing. If I move it a bit more, now I'm like here on this line. And if I move it all the way, I'm now, my 2, 4 split has gone away actually. I'm just left with my 1, 3 split. Okay, so this line here is all trees I get by adding leaf 4 somewhere along this orange edge. And now I can just do the same thing with my other 4 edges. So to add it along the green one, right? So this is it. So I've only got the 1, 3 edge, I'm right here. And notice they connect over. And now as I move it along, I would move to here, right? So I've got my 1, 3 split, but it's like half the distance it was. And I've got this new 2, 4, 5 split, which is this one. And I just realized actually my trees did switch to being unrooted, but it's all okay. And then finally, so as I move this along, like now I only have the, so this 2, 5 or 2, 5 split here, right? That's that full edge and down here. So as I'm adding this edge 4 along this green line, going from this point to this point, like each different place I could add it in is corresponding to one place, one point along this line. Okay, so then I have my other 3 edges, and so each of them has a new orthant for the new tree shape or topology I get from adding leaf 4 on that edge. And then the exact possible trees I can get are going to be the ones given by these dark lines in the space. So you notice in this, right, that the extension space, so the space of possible trees we get by adding leaf 4, looks like this tree here, which is kind of not surprising because I can add leaf 4 like anywhere along here. And there are actually sort of 4 other dimensions for each of the edges to the leaves, the 4 here plus leaf 4. That I'm just not showing in the picture. But they would actually be important in doing the computations. Okay. So that's fine. So the paper by Wren et al., they talked about exactly which orthants you can have, which of these new orthants you can have. I mean, adding the one leaf, it's not so complicated, but if you think of adding, you know, some number of leaves, it's like a lot more complicated which orthants you can get. So they showed that it's you want to increase it to have capital N leaves. Your current tree has small n leaves. And so this expression here is a number of new orthants where your double factorial is your skipping ones as you go down. So this is actually the number of possible phylogenetic trees on capital N leaves divided by the possible number of phylogenetic trees on lowercase n leaves. So this could be exponential, right? And quite badly exponential. What we would hope is that most of these trees are only missing a few leaves, if any, and so it would not actually be completely crazy. And then we can find within an orthant, we can find so if I'm in the orange orthant, say, I can find this region by solving a system of linear equations. And these are actually all the equations for the different, so what we want to do is if I'm adding leaf 4 somewhere along here, I just need to make sure that or maybe let's think about it along the green one. So if I'm adding leaf 4 here, I just have to make sure that the 2 new edges I make add up to the sum of the old edge, right? And that's why I'm getting this like diagonal line here. And obviously if I'm adding more leaves, like maybe I add like several leaves along here, I would just have to make sure that the sum of all the new edges add up to the previous old edge. Okay. And that can be encoded, right? So I'm just saying like, if this edge, the first one is actually for the green edge, right? So I'm just saying that the sum of the 2 new edges I make have to add up to the previous old edge. Okay, and that would be, so here we actually have each equation is for each of the 5 different orthants, what this line is. And if you're adding more leaves, like these are a little more complicated. And in general, I can think of there being a 0-1 matrix that's encoding which splits in the extension space, so which splits on capital N leaves come out of which splits in my original tree, right? So for example, I would be encoding that this split 1, 3, 2, 4, 5 came out of this green edge, and same with this one came out with those edges. So those, like, you can kind of think of all the edge, let me just think the ordering. The columns are my original edges. Sorry, are my edges in this, like say the orange orthant. The rows are the ones in my original tree. I think that's it. And then I have a 1 if one split is going to the other. And then times your edges in your new orthant have to equal your edge weights in your original tree. Okay.
So, sorry, Megan, I have a quick question.
Yeah.
Yeah, so obviously you've written down the equation for a polyhedron, at least when everything is in Rn. But am I solving this equation in tree space, or?
No, so that's actually the kind of the trick, is you are solving it individually for each orthant.
Nice.
So well, nice and not nice. So I mean, within each orthant, yeah, it's just a linear system. That's great, it's very fast. But we said that there's potentially exponential number of orthants.
Mm-hmm.
But then my next caveat was that hopefully in actual examples, that's not the case, because we don't have random trees. We have trees that hopefully have something to do with each other and are not missing huge number of leaves. So yeah. And I should say all the details, like, I mean, our paper is out now on this, and it has a bunch of notation that I'm kind of like sweeping under the rug, just because I didn't think people wanted to see it. I thought people would like pictures better. So I'm trying to give kind of the overview and intuition of what's happening. And then if you want the like gory details, then they're all in the paper. And it's all worked out like, precisely as it should be.
And what's the title of that paper, just so?
It's the same as the title of this talk.
Okay.
The representations of partial leaf sets of phylogenetic trees. And it's in SIAM the CIAGA Journal Sociam on
Yep.
Journal on Applied Algebra and Geometry. And I'll have the reference at the very end of the talk too.
All right, great. Thank you.
Yeah, so what we were really interested, right, is having multiple trees. So here I just look at 2, but obviously we would, in general, we consider more trees. And they're each missing different leaves. And we want to kind of look at, like, can we somehow put them together to make a big tree with all the leaves? So the T1 is a tree with seed, and we saw we could add leaf 4 and get this one in bold. And tree 2 is missing leaf 5, which if I add it, it looks like this. So again, right, you can see it's the outline of the tree roughly, where now this middle branch has 1, 3. So if I add along the middle branch, right, I get 1, 3, or if I add, or 2, 4, which is now here. So that's by adding leaf 5. And then I can think about what are the 2 together and where do they intersect, because that would be the tree that is sort of a super tree that encapsulates both of these trees with missing leaves. And that's great. In this case, great, they do intersect at this one point, and we can all be happy. Now, if you know, I think probably some of you are starting to see the problem here, because, right, remember that this is really only 2 dimensions out of 7. So here it kind of looks like no matter how these trees got moved around, they would always be intersecting, but really I have 7 other dimensions here. Actually, so we'll get into that question in 1 second. So let me just talk about the general one. So in general, for each of my trees I can have this matrix encoding how, a matrix for each orthant, right, encoding how the edges in this new orthant collapse down, you can think of, or map to edges in the original tree, 0 matrix, my solution in my new orthant, my original edge lengths. And so I'm, again, for each orthant, I'm just solving a system of equations, which now might have no solution. So for example, you can see these 2 orange lines don't intersect. So there would be no solution in this orthant, but there is one in this orthant. Okay, but we can still solve this pretty fast, or quickly, right, and just check all our orthants. The bigger problem is that it might not be robust. So let me just walk through this. So again, I think if you have familiarity with doing this kind of thing, you're immediately seeing it. But if you don't, let me show you, because it's a huge problem. So let's just imagine this edge 2, instead of being 0.35, it was 0.3. Okay, so now my 2 equations for my, and I'm just worried about the green line, because those were the 2 that were intersecting, right? Was if leaf 4 was added to this green edge, and leaf 5 was added to this green edge. And so the 2 equations I care about are these 2, that orthant, and these 2. And when I actually map them out, I get this situation. Now I'm looking at 3 of the dimensions, and these 2 lines, there's a gap between them, right? So there's actually no solution. And so just by wiggling, and I'm sure I think I probably just had to make this even slightly smaller than 0.35, like 0.35 minus epsilon, right? And I immediately get no solution. Okay, so this is like not robust, right? Because obviously there's error in getting these edge lengths in these trees. So if everything has to work exactly to get your answer, this is not any good. So we have a solution. And that is what you want to do, is think of there being around each of these edges. You want to think of there being, or sorry, each of these, the possible trees that exactly come from it, which are in bold, they're sort of being this alpha relaxation. So that the edge lengths could actually differ by either alpha, plus alpha, or minus alpha. So the equation for this one line becomes 2 equations. And unfortunately, let me just see if I can move. Okay. So, and unfortunately, yeah, so so the original weights, I'm allowing a difference of either plus minus or minus alpha, or the original weights to instead be kind of alpha less than they should be, or alpha more than they should be. And I can set this up as a linear program. because what I might be interested in is if I have a whole bunch of these regions, so I can make one of these regions for each of my trees, and I might be interested in what's the minimum alpha to find a solution in an orthant, right? Because that's some measure of how far apart the two trees are, or sort of how compatible the set is. And so I can set it up as a linear program. Again, I still have my sort of 0-1 matrices doing the encoding, my solution in that orthant, right? And again, this is still an orthant by orthant situation. And again, there's still a hidden issue that in the worst case, there are an exponential number of orthants. These are my weights in my my original trees. And then what I can add in is kind of this, I can think of YP is sort of an error tree of positive values that I'm adding to it, and negative values I'm adding into my tree, sort of the error bits. So anyway, so you can set this still up as a linear program to solve, so that's can be done quickly. And that is more or less where we stopped our work. So I would not say, we gave another method that was proportional for a way of relaxing this. But really, I think this paper just kind of gave more questions than it basically opened. There's a bunch of open questions here, is what I'm trying to say. Neither me nor Jillian are currently working on any of them, so they like are sort of truly open in that sense. So, and we have them in more detail in our paper, but let me just kind of talk a little about them. So one idea is, can we somehow quantify how much incompatibility there is in a set of trees using that relaxation parameter and like figuring out the minimum? And then the other idea is, like, could you even set it up to be some kind of hypothesis test? We did think about it a little. There's nothing completely like super obvious, but I mean, for sure, I think there might be like, I think this is an important idea of like, could you somehow qualify exactly how far, like how much incompatibility or how much a set of trees sort of came from some super tree or not, like how much adjusting do you have to do to each tree to get it, or how much relaxation do you have to do. Right now, none of the theory was extended to non-binary trees, which do show up in biology. And then this is maybe a bigger one too, whoops, is that, is that right now we don't allow topological error. So our alpha relaxation was only allowing, within each orthant, only allowing the sort of edge lengths to change. But for example, we don't, so for example, from this axis, right, remember, oh no, I have all my 3 quadrants from it. Let me think. I mean, so essentially we're not allowing some, if say, let me go back to the picture. No, I guess I have 3, okay. So we just aren't allowing, like, essentially we're only allowing the topologies that show up in these, if I look at these union of extension spaces, that's the only topologies we're allowing in our final tree. But like there can be for example, there can be topological errors in these trees. Like if instead, which edge is this? Yeah, like if, like this could be, for example, like if this green edge was really small, both of these 2 points would be very near here. And so you could imagine that maybe instead you'd actually get a different, it should really be a different tree. It should be, I don't know, like 3 and 5 paired together, something else, right? So we're somehow not allowing any topological error, which we know happens in real data, that the gene trees can have errors in their topology. And so I think that would actually be a really interesting and important area to extend this, is to somehow allow some other topologies beyond just the ones that sort of appear in these extension spaces of the trees, of your input trees. Okay, so thank you very much. This is, so here's the paper that this was all based on. And yeah, and then also my webpage if you want to see any of the other papers that I talked about.
All right, great. Thank you, Megan. And I'm just going to go ahead and encourage everyone to give a virtual round of applause to Megan. So I'm now available to take questions. Again, if you have a question, please just put it into the group chat for everyone, or you can unmute yourself now. Maybe I'll just give a few seconds if somebody wants to unmute themselves. All right. Well, maybe just to get discussion going. So Megan, you mentioned the relationship between the sort of relaxation and the concept of sort of reach. So should I be thinking of, every one of these trees sort of embeds into the space of trees where you can look at the possible one branch extensions, and then you're kind of taking like a tubular neighborhood about sort of two of these, and you're asking what's the sort of smallest value of alpha that those intersect?
Mm-hmm. Yeah,
right.
Yeah, that would be one way to think about it. Yeah, that's like, sorry, I like, yeah, so I've only got the one tree here, but what you'd really, maybe it's easier to think of this picture, right, is that instead of having just only the extension spaces for the trees, you'd have those tubular, like that kind of alpha. Yeah, you'd be thickening these kind of by alpha on each side is a way to think about it. And again, this picture is slightly misleading because of course it looks like you don't need, no matter what, they would intersect. But we can see, because of this robustness, you can see that if they, like there's right now this little gap between these two lines. But if they were thicker, they would then be intersecting. So it's essentially taking care of this issue with robustness. So within the orthant, that just by shifting one of the edge lengths slightly, because you're really working quite high, like higher dimensions, your two lines can be like a little off from each other.
Right.
And so then as soon as you make them, because now there would be like in the 3D picture, right, there's like a 3D tube around them. And so now there actually would be you could, assuming these two green lines were sort of close enough, right, there'd now be a point where the two tubes intersected.
Right. And then if instead of just extending by a single edge, you wanted to try to like add two edges, then when you would sort of now have like a two-dimensional space embedded, and maybe there's more likely to be a two branch extension, or am I thinking about this wrong?
Yeah, so I mean, it would be the same thing that each of the new edges would have like I mean, you now, like for each, let me see. Well, okay, so no, I guess you're only changing the widths of the original. So if you look here, you can see these are the W's are the widths of the original branch lengths, and those are what we're allowing to differ. So it wouldn't. Yeah,
I guess that wouldn't. Really
yeah,
change.
I guess like I must say I've not actually thought about how to visualize this. Like we just kind of did the one leaf, and then we kind of worked out the equations for it, and then we said, okay, like how do we generalize those? And then we did all our generalization more or less with equations and not trying to visualize it.
Yeah, no, it's still really fascinating. All right, I think that's it for my questions. Does anyone else have a question? They can unmute themselves.
Hello, this is Seth. Yeah, I'm wondering how, from the application of the biology point of view, what questions would your methods apply? In the researches of the biologists.
Yeah. I mean, that's a good question. It's like how much, I mean, like exactly how much of, I mean, of course I can always be like, oh yeah, there's these applications, and exactly how much carries over. So right now people do look at super trees methods, but usually I think the ones that are actually in practice are pretty much they're looking at topologies and how to fit topologies together. There's actually, yeah. So I don't think there's a lot of work with branch lengths. Now, a lot of biologists will kind of justify this by being like saying, oh, well, you know, there can be lots of errors in branch length, so why should we, like, we're going to throw this information out. But I mean, even though there are errors, I mean, you still have information, especially say relative, like, I mean, so yeah, right now, I don't think this is, I mean, it's going to in any way help biologists. The idea is that hopefully we're setting up a framework and maybe as more, like, it's a framework to think about it, and hopefully as there's more work in it, it might.
Yeah, of course.
I think, yeah, so I think some of the reason why you might care about compatibility is to try and figure out if you're trying to find, so with the gene events, some of them are more common or rare than others. So yeah, so that's like, so for example, the incomplete lineage sorting, that happens all the time. For example, if you've ever seen data on human, chimp, and gorillas, you'll know that I think about, I want to say like 1/3 of our genes have, so if you take genes in all 3 species, that 1/3 of them have chimps and humans and gorillas, I think, paired up instead of humans and chimps. And that those differences are actually all can be explained by the theory, this incomplete lineage sorting theory. So that's really common. The horizontal gene transfer and gene duplication and loss are less so. And so, and sort of detecting like horizontal gene transfer is kind of a harder problem. And so perhaps if you could talk about compatibility, I mean, like maybe you're going to be able to say like, well, like maybe you're going to be able to tell if like if there's not a lot, if like within a gene set, it seems to have less compatibility than some other gene set, that's some indication that you have these other like evolutionary events that happened.
So I get the sense that the biologists obtain collections of different trees. Each tree is subject to some inaccuracies in both a lot of inaccuracy in edge length, and a smaller amount of inaccuracy in topology. So the basic data that they wish to process is a collection of phylogenetic trees.
Yeah,
they know maybe up to some error which leaves correspond to one another.
Yeah, so that actually they would always, well, not always. So I mean, in a traditional application where your genes are coming from some species you collected, you do know which gene came from which species.
Yeah.
There are other applications, though, I think, of this where it's less clear how to map. So, for example, with the trees from epidemiology, your leaves are actually usually, they've sequenced a virus from some patient, and so and so there's this time element, right? Because obviously people get diseases over different periods of time. Yeah. And also it's not like, so now if you want to compare, like, how does the trees, say for measles compared to that for Ebola, well, how many people have had both measles and Ebola, right? Like, so you don't have this common species that had both genes. So there's some other, like in other areas other than sort of evolutionary biology, sometimes you have this issue of like which leaves, like if you have 2 trees, how can you match up the leaves?
Oh, that's also, yeah, that's another issue. Yeah, I see.
Yeah, which is a whole other issue. And I mean, this actually might be relevant to, we mainly focus on the evolutionary biology problem.
Yeah. But basically the data to be analyzed is a collection of trees, and you want to find relationships, similarities, pathways between the trees. Okay, good.
So Megan, just to follow up on that question real quick. So has there actually been sort of, say, let's like fix a tree topology or a collection of tree topologies. Have there actually been empirical studies of like what is the empirical distribution on the space of possible tree topologies?
Yeah, so there was some stuff. So there was a student of, oh, I don't remember who he was a student of, Zacharias. The paper I mentioned by Zacharias et al. He was a grad student at, well, he was an MD PhD at Columbia University, and he had looked at, I want to say from flu vaccines. So he had not looked at gene trees, but he had looked at these, like, because it was flu, right? You have this issue that like each thing you have is from a different patient. So somehow it's harder to match stuff up. But he had looked at, I think, 4-leaf trees. So we just would sample 4 patients, construct the tree,
right,
or 4 virus samples, construct the tree, and look at those. And he looked at how those are spread out over tree space.
Yep.
And he had some conclusions, which I must say I don't remember. But that would be, yeah, his last name is Zaris. And he has 2 or 3, he has at least 2 papers I can think of. There might be more.
Okay.
Yeah, so he had looked at some stuff of like sort of like actual data. How does that, and actually visualizing it spread out over tree space.
Oh, fantastic. All right, thank you.
So I have a question about this example of the Peterson graph that also showed that it was not Euclidean.
Yeah.
So if you add more species, these graphs are understood, like more leaves, or like...
Yeah. So these are, so the Peterson graph, right, is made up of these pentagons, right? Like the star is a pentagon, and there's pentagons all over the place. So these pentagons are actually associahedra.
Okay.
So as you go higher, what you have is you have an overlap. So I think if you go up 1 dimension, your associahedra now have it's a polytope with 14 sides, I believe. And each orthid or combinatorial tree topology, right, is in multiple associahedra. So like this one, right, is in this pentagon, in this pentagon, in this pentagon. Yeah. So so as it generalizes, you have these overlapping associahedra. The original paper by Valera, Holmes, and Vogtman talks about this a little. I think they kind of do out the calculations, right, of like how many associahedra is each of these tree topologies in. So each associahedron is corresponding to an ordering of the leaves. So like this outside one, I think I fixed, if you think of your leaves as always having the ordering A, B, C, D, then these 5 trees would be all trees with that ordering, right? Like that's one definition of associahedra, right? Is you have these trees and you fix the order of leaves, and then the associahedra encapsulates all those trees. And then, but I can, from a phylogenetic point of view, the ordering doesn't matter, right? Because you just care that A and B are most closely related. It doesn't matter. I could write B and then A. And then that's how you get that this tree is in different associahedra, because it's in all the associahedra with each ordering it can have.
Okay, thank you. Great.
So, yeah, there's some really interesting combinatorics there that I'm kind of like, I didn't talk about, but yeah. And the other person actually who's done some stuff with the tree spaces and the combinatorics from a more topological is Satyan Devados's work. I don't know if any of you know him, but he's done a lot kind of at the, like looking at sort of the topology and the combinatorics of kind of extensions of this, or extensions or variations of this tree space. Probably not strictly extensions or variations. It's some moduli space that he's looking at that's related. And he looks at graph associahedra, which are kind of like some extension of associahedra, I think, to graphs instead of trees, and sort of spaces coming out of those and stuff.
Yeah, I had a question of elaborating on my previous question.
Yeah.
To what extent do the biologists actually have accurate observations of the trees themselves? Or is it that the biologists simply have data about each leaf and distance information between pairs of leaves, and then they reconstruct the trees from that?
Yes. Yeah. So they're reconstructing the trees. So already the trees are actually like an like an output of
Mm.
an inference method. Yeah. So their data is the DNA sequences. So yeah. So either you can measure distances between the DNA sequences, and there's ways to make trees from that. Usually, they're making trees. There's different. You can model evolution as a Markov model. And so that's what the sort of most popular programs now use, is they're either doing Bayesian inference or statistical inference, but with this like underlying Markov model, and then trying to infer the best tree that fits the data.
I see. So there was the fundamental, the concrete experimental data consists of a collection of DNA sequences, and then there's one of the current techniques for deducing the tree is to simulate Markov models and search for optimize the fit of the result of a model with the data.
Yes. Yeah.
Okay.
And then the problem is that you basically, because the number of trees is huge, that the problem
so they also want to so they also want to model how the collection of the raw experimental data is a collection of collections of sequences, a collection of DNA sequences. They want to model how that object how that object came into being by evolutionary processes.
Yeah. And I mean, there's like other problems, like, for example, you have issues of you have your genes from your different species, but how do you know how to, like, all the programs for making trees assume that your genes, you've aligned them so that you know, like, which, like, I don't think I do have a column of them aligned, right? So
yeah.
If you've ever seen DNA, right, you see it often, like, the idea is like this letter here was all coming from the same original letter. Right? And that this G, these are all from the same original G. But of course, there's like, this looks like a very nice species, but usually there can be missing, like, just like there can be missing genes, there can be missing like base, like, letters or extra letters. And so that's even a whole problem there of, like, this whole question mark in between the DNA and the tree, like, there are that's a whole.
Okay. So there's more structure to a collection of DNA sequences than merely sequences that you.
Yeah, because you kind of want to know, right? If you want to look at, like, how did they evolve from some ancestral one, you have to have some idea even of how to, like, match, like, what parts were from the same part. So and then there's even, so I guess there's maybe three problems, because there's that sequence alignment problem. There's how do you get from the line sequences to the tree? And then even before that, there's how do you get from the DNA molecules to the letters? And there's actually, that's a graph, or what are those called? De Bruyne sequences. There's a whole bunch of stuff with De Bruyne sequences.
Yes. The what's extracted in the laboratory are subsequences.
Exactly. Yeah, you have all these little pieces, and then you have to, like, sort of assemble them into your long gene. Yeah. So I mean, I'm just like, there's a whole pile of other, like, stuff there that I'm not.
I would think now that the strategies to discover the trees can be disrupted by what you call it, cross exchanges.
Oh yeah, like the horizontal gene transfer, right?
Yeah,
like
the horizontal gene transfer. Yeah, that because the structure is not a tree anymore.
Yeah, no, exactly. And so then there's also an area where people look at these as networks instead of trees.
Yeah. Okay.
Which, like, and for mammals, a tree is fine, and then for bacteria, you really for sure want to do a network. Yeah, right. And then for different, like, I mean, there's other things between bacteria and mammals, and then some of those you should maybe also be looking at networks.
Yeah. Okay, cool.
Yeah.
Great. Well, just to try to simulate a real talk, it's 10:04, and so
Sure.
I don't want to infringe on anyone's coffee break time. So maybe let's give Megan one more round of applause, and then the next talk's with Francois at 10:30. So.